Hello, BNG411 class. This is the pre-lab, the recorded pre-lab lecture for next week's lab, starting the 26th, on the separation of proteins by SDS-PAGE. A couple of things to note. First is that I'm not around next week, so this is why I'm recording this and posting it on my courses. So please watch this before your designated lab. And also, please note that if you are going sequentially in the lab manual, then you will see that up next in the lab manual is supposed to be enzyme assays. We're going to do those later on the lab schedule that is in the syllabus that you all have read. SDS-PAGE is what's next. So SDS-PAGE is the lab you will be doing this coming up week, this next week, starting Tuesday the 26th. Okay, so let's talk a bit about some methods of protein separation. So, when you have a product that you want to recover from a cell mixture, from some growth media or some other mixture, there are a variety of ways that you can separate different proteins based on different characteristics. And we'll talk about quite a few of those over the course of the semester. So we can separate them by centrifugation, and we will be doing some centrifugation later. We can separate them by filtration, by some kind of filtration, gel filtration or chromatography. We'll do that also later on in the semester. And we will separate them by SDS gel electrophoresis, or SDS-PAGE, as it is known. And that's what we're going to focus on this week. And so there are ways to separate proteins based on the characteristics of those proteins. You can separate these proteins by size. Obviously, big proteins will filter out differently depending on the size of the pores of your filter media, differently than would smaller proteins. You can separate by charge, so you can apply a charge to some media and be able to separate proteins in that way. You can separate them by shape, and that's a lot of what differential centrifugation is about. You can separate by density. If the proteins bind something, if they're enzymes and they bind a substrate, you can separate the proteins in that way, and that can end up being very specific. Or you can do some combination of these. And if you are purifying proteins as a job, you probably would use some combination of attributes for your protein in order to separate it from the rest of the cellular proteins that might not be so valuable to you. And so we'll talk a little bit about centrifugation. We're not going to do any differential centrifugation in the lab, although it would be nice. But differential centrifugation is a little bit different than just spinning things down in a tube and removing the supernatant. Essentially what you're doing is you're taking a cell lysate, you've taken your cells from your culture and blown them apart, and you have all the proteins. And what you do is you spin them at different speeds or in different types of media, and cell parts of different densities will pellet on the bottom depending on what you're doing. So if you have a low speed centrifugation, essentially what you'll bring out is all the whole cells that didn't get blown apart. If you're working with eukaryotic cells, cell nuclei, and even cytoskeletal elements will get pelleted if you do something at low speed, leaving in the supernatant a bunch of proteins and other smaller organelles and complexes. You can do a higher speed centrifugation and then pellet down all the mitochondria and other types of organelles that are smallish. You do it at an even higher speed. You pellet microsomes, small vesicles, things like that, and you're left with protein complexes and other macromolecules. Spin it at very high speed, you can get those out as well. So essentially what you've done is you've separated large protein complexes and even whole cells from soluble proteins. And you can certainly do that, and there are a variety of ways to do that that we won't get into. And you can do isopycnic centrifugation, which is a little more of a one-pot type of method, where you have what's called a gradient. So you have a higher percentage of, and usually it's sucrose, but you have a higher percentage on the bottom, a lower percentage on the top, and then you carefully input your protein complex, in this case is ribosomes, and you spin them at a certain speed, and based on the gradient and how fast you spun them, the smaller molecules will float up to the top maybe. You'll get the medium-sized molecules in a band in the center, and the larger sized molecules you can find down at the bottom towards the tube bottom. So you're using a gradient of a solution to separate particles that have different densities. And usually this is sucrose, although in the old days and with different sized pieces of DNA, often cesium chloride was used, a cesium chloride solution. And again, this works best with large protein complexes like ribosomes. So, kind of the standard illustrations and standard techniques focus on things like ribosomes. And then there's what we're going to do, which is the SDS-PAGE. Now, what does SDS-PAGE stand for? Well, SDS, as I'm sure those of you that remember BNG411 know full well, SDS is sodium dodecyl sulfate, which is a detergent. And this is what SDS looks like, the molecule. You can see it's an ionic detergent. It's got this nice long hydrophobic tail, and it's got this charged head group with a sulfate, and that's complex to sodium. So that's what sodium dodecyl sulfate looks like. And then the PAGE part of it is polyacrylamide gel electrophoresis. And what we're doing is we're separating proteins by and large by size or molecular weight. So we have proteins that we will coat in the detergent SDS, and then we're applying them to the gel here. We're putting them in this little well, applying an electric field, where the negative is up here, the positive is down here at the bottom of the gel, and that's the direction that the migration will go. And then once they've separated by size, and keep in mind polyacrylamide gel is kind of like a little meshwork, so the longer the protein is, the higher molecular weight it is, the longer it's going to take to snake through the gel in the direction of the electric field. So the larger proteins will be towards the top, middle-sized proteins will be towards the middle, and the smaller proteins will be towards the bottom. And here's what a setup might look like, and it's kind of like the setup we'll use in the lab, where we have the gel here. It'll be sandwiched between two plastic frames. You'll have a stacking gel, more on what the stacking gel does later. Sample wells in the stacking gel will allow you to put your sample in, your treated sample. And then there's the running gel, which is what most of the gel is made out of. And then this is bathed in a buffer. So this buffer is a special, what we call running buffer for the gel, and it's got some salts and some buffers in it, some actual buffers in it, and this allows for good transfer as the current goes down and the proteins go along with it. So we're going from the cathode to the anode here, and that's the direction of migration of the proteins. So why do we use SDS? Why do we bathe our proteins in detergent before putting them on the gel? What possible purpose could that have? Well, first of all, if you're doing regular old SDS-PAGE separation of proteins, you're going to want the proteins come all balled up when you take them out of the cells. You're going to want them in nice linear strands so they can snake through that polyacrylamide. So, you want to break up that secondary structure, the tertiary structure that proteins have, and coat them with a negative charge, which we'll talk about more because it's important. We'll talk about that later. So essentially, before SDS, you have a bunch of interactions, hydrophobic interactions, charged interactions, and salt bridges, and you have hydrogen bonding and things like that make up the protein's overall structure. After you treat them with SDS and denaturing agent, you have these linear chains of peptides, proteins that are coated in a negative charge. And again, why PAGE, polyacrylamide gel? Why the gel matrix here? Well, it is because it's simply a matrix. It's kind of looks like maybe a sponge or, you know, a big mesh of polymer, and there are tunnels of different diameters and different lengths, and it's a substrate for the proteins to snake through. So we have, you know, proteins will enter these tunnels or whatever, and they'll snake through. The smaller will go through quicker because they're able to, because they're smaller. The larger will take longer time to go from negative to positive, and that's why they will end up at the top of the gel, because they haven't gotten as far. Okay, so how does this work? So essentially we have treated our samples with SDS, we've denatured them, we apply the protein with dye so you can follow the direction the cells go, the protein goes. We apply them to these little wells in the gel here with a pipette. You know, and this is in the stacking gel. And then the bottom part of the gel is what's called the resolving gel. And so we run the electrophoresis. So we put a current on them, and the current is, you know, we go from negative to positive here. So proteins are going to migrate down the gel from negative to positive. And you can follow the dye that's in these samples in the form of a dye front as it goes down through the gel. And you can see how far the gel has gone based on that dye front. Then we can remove the gel. We would stain it in something called Kumasi blue. You might remember that from our protein detection and quantitation lab this past week. That will, you know, solutions that have a lot of protein in them will turn blue. Well, it's the same sort of effect here in the SDS-PAGE gel, where we stain them with this Kumasi Brilliant Blue stain, and the blue will complex with any protein in the gel, and then when you destain, so remove the stain that isn't binding any protein, when we destain we can see where the proteins have migrated to. Okay, so a little bit about the buffers we'll use. You will really only run into one of the buffers, which is the running buffer. It's basically what's called a tris-glycine buffer. Tris is the buffer system that's being used, and glycine is an amino acid. And more on why those are important in a bit. But if we were to make the gel, and we're not going to make the gel, you're going to use pre-made gels. If we were to make the gel, you would use what's called a discontinuous buffer system, which means you have different buffers in the gels and in the tank, and you can see that they vary by pH here, where we have pH 8.3 in the running buffer, pH 6.8 in the stacking gel, and pH 8.8 in the resolving gel. The stacking gel is usually of lower density, and the separating gel is of higher density, and this slows down protein migration. But you might ask, why the different pH values? So the running buffer and the running buffer in the tank is tris-glycine. Glycine, as I said, is an amino acid. At pH 8.3, glycine is negatively charged. And so you turn on the power to run the gel, glycine from the buffer must enter the stacking gel because it's negatively charged. We're going from negative to positive here. So a negatively charged molecule is going to want to go in the direction towards the positive charge, so down towards the bottom of the gel. At different pH values, you can see glycine will take different forms. So at low pH, it'll be positively charged. We don't have to worry about being at low pH. We're going to be either at neutral pH here, where it's a Zwitter ion, that's in the stacking gel, or it'll be a negatively charged ion at high pH, like the pH in the running buffer and in the resolving gel. So as I said before, in the buffer reservoir and in the separating gel, we have a glycine ion, a negatively charged ion at at high pH, because the buffer that makes the separating gel and the buffer in the reservoir are of relatively high pH. In the stacking gel, you have the Zwitter ion, which means it's positively and negatively charged, and those cancel each other out in the stacking gel. Okay, so what happens in the stacking gel? Well, glycine has become a Zwitter ion. It's neutrally charged, or charges cancel each other out. So now it's going to move very slowly. Doesn't have the same impetus to get from the negative charge to the positive charge. So it's going slowly now. Chloride ions as part of the tris buffer, the tris HCl buffer, the chloride ions are moving faster. Fast-moving chloride is going to pull slow-moving glycine along. And the proteins have intermediate mobility somewhere between fast-moving chlorine and slow-moving glycine. So they're going to be somewhere in the middle there. They are concentrated in the zone between these two buffer ions, or to be clear, the chloride ion and the glycine Zwitter ion. So that's going to allow them to run very slowly through the stacking gel. And then they're going to hit the separating gel. The proteins will be dumped in a very narrow band between the stacking and separating gels. So basically none of the proteins will really and truly have what we would say is a head start in getting into the separating gel. We want all of the proteins to enter the separating gel more or less at once, so they can truly be separated by size. So again, at pH 8.8, which is what the separating gel is, glycine is now negatively charged, so it's going to speed away towards the bottom of the gel where it's positively charged. Proteins are coated with SDS. They're negative, so they're going to start to separate and migrate towards the bottom. There's a big impetus for the proteins to get down towards the bottom. The only thing that's preventing them from doing so at this point is the mesh work of the acrylamide, the polyacrylamide that's in the gel. So essentially what's going to happen, the proteins will be loaded onto the gel, you'll turn the power on, current will flow, proteins get bunched up in the stacking gel, and then proteins will separate based on size in the separating gel, or the resolving gel, or whatever you want to call the bottom gel. And here's what this looks like again. Again, your loading sample. We don't have apparatus that look like this, but the idea is the same. You have the cathode up here and the anode down there. They're going to migrate towards the anode. So you load your sample. That's the direction of migration down to the bottom. That's exactly how our gels are going to run. They're going to migrate down to the bottom, the negatively charged proteins. You have your protein mixture. This would probably be in the separating gel. In going through this porous gel, they're going to separate based on size. So when proteins leave the stacking gel, they'll be up here and in the separating gel. By the time you shut it off, hopefully the proteins will look like this. Okay, so what am I looking at? How do I know what these proteins are? They're just a bunch of bands to me, right? Well, what difference does it make? Well, you figure that out based on the molecular weight marker here. So we will also load in our lab, we will load the molecular weight marker so we can keep track of where things are. So in this molecular weight marker, we've got like 100 kilodaltons down to 3.4 kilodaltons. So it's a pretty good size range for most proteins. And then you can look at your samples and see how big or small are your different proteins. So you can see there's some that are around 45 kilodaltons, 40 kilodaltons, 28.9 kilodaltons, things like that. So you know where things have migrated to based on determination by where your molecular weight marker standards have run. Okay, so let's see if we can run this video here, video within a video, of SDS-PAGE in action. And so it'll give you an idea of what you're up against.
Samples and markers are loaded into wells at the top of the gel. A running buffer is added. As electric current runs through the buffer, the negatively charged proteins migrate towards the anode. The polyacrylamide gel contains a stacking layer on top of a resolving layer. The stacking layer, which has a percentage T of around 4%, contains much larger pores than the resolving layer. Proteins tightly stacked like coins in order of decreasing mobility migrate through the stacking layer. At this stage, mobility of the proteins depends on the electrophoretic conditions. Because of the drastic difference in pore size, once the protein stacks reach the resolving layer, the proteins begin to separate according to size. Because small proteins experience less resistance as they move through the pores, they move more quickly than larger proteins. The dye front consists of a very small dye molecule, like bromophenol blue, which is mixed and loaded with the sample proteins. Because of its small size, the dye front moves ahead of the proteins. The position of the dye front is a good indicator of how far the proteins have progressed through the gel. Electrophoresis is complete once the dye front reaches the bottom of the gel.
Okay. All right. So question here. What if I'm looking for proteins on a nicely run gel like this one here, and a couple of them are relatively the same size? Like, how am I going to know? How am I going to know that they're both there? And you can see there's a lot going on here. There's a lot of bands of proteins in some of these wells. So how do I know what's what? Well, we can run what's called two-dimensional gel electrophoresis and separate the proteins by two attributes. And we're not going to do this in lab, unfortunately. We don't have the apparatus for it. But essentially how that works is you're going to separate them first by what's called isoelectric focusing, or by their isoelectric point, which has a lot to do with what their charge is, and then separate them by the traditional SDS-PAGE via molecular weight here. And there's a video that I will post on my courses that you can watch if you are so inclined to do so. You don't have to, but if you're interested to know more about 2D gel electrophoresis, you might want to. So again, 2D gel electrophoresis that separates proteins by two different attributes, like I said, charge and then size. So first you run what's called the isoelectric focusing gel, then you run traditional SDS-PAGE. And so then you see proteins are separated by charge, and then finally by molecular weight. Well, maybe we'll get this video after all. Let's see what we come up with here. The video is unavailable, so we'll come up empty-handed on that, and that's fine, because we're not really going to do much with 2D gel electrophoresis. Okay, so again, isoelectric focusing, back to that, the first step in 2D gel electrophoresis here. So at low pH, most proteins have a positive charge. At high pH, most proteins have a negative charge. And when an electric field is present, each individual protein will be pulled towards its isoelectric point, wherever it's got a neutral charge. So essentially, your what's called your isoelectric focusing gel will have a low pH at one end and a high pH at the other end. And sending electric field through it is going to separate these guys in terms of where they feel most comfortable, where they're neutral in charge. So again, you can have your proteins distributed throughout the gel. The current off, they're kind of randomly distributed here, but once the current is on, the proteins will migrate towards wherever they're neutral. So pH 3 would be down here towards the anode, pH 9 and 10 would be down here towards the cathode. And here are some isoelectric points of several common proteins, things like pepsin, ovalbumin, serum albumin. Pepsin's really very acidic in terms of its isoelectric pH. And the reason for that is it's found in the stomach. Whereas you go down here, things like lysozyme, histones, cytochrome C, they're more basic in terms of their isoelectric pH. And this might have something to do with where they can be found in cells. Okay, so now that you've separated proteins by isoelectric focusing, or separated by charge, we do the old SDS-PAGE to separate by molecular weight. And then what we can do is we can, based upon charge and molecular weight, we can now potentially identify different kinds of proteins, because we can predict their molecular weight based on their sequence, and we can even predict their charge based on their sequence. So we can pinpoint where they might have ended up. And here you can see this is a 2D PAGE gel that has been run, and they've identified several different types of proteins. And so we don't have to use SDS in our PAGE gels. Say we want to keep our proteins intact. Well, SDS-PAGE is not going to do that for us. It'll denature and it's going to separate proteins. Native PAGE, however, will keep the protein structure intact. So essentially you end up separating by size, shape, charge, etc. And you saw the crisp bands that could be made with SDS-PAGE gels. The bands in native PAGE are much less what we would call clean. They're more blurry like this, but the proteins in them are folded still. They have their native conformations. And you could potentially remove them from the gel and have a separated, purified sample of your protein in its native form. So again, no SDS is added, no denaturation, no covering with negative charge. It helps to have an awareness of the isoelectric point of your protein, and that because that's how you want to choose your buffer system. That's the critical thing, whether it's acidic or basic. Different buffers with different pH values will affect the interactions and the mobility in native PAGE gels. So you can see certain buffers have certain effects, where with this protein, whatever it is, it looks like it's a purified protein, has possibly dimerization going on in either phosphate or citrate buffer. And whether that's what the authors of the study wanted, I don't know. But this is basically an illustrative of the different interactions that could occur depending on whether you're using an acidic or basic buffer. So for example, we could look at diphtheria toxin proteins. We can compare SDS-PAGE and native PAGE. So here's SDS-PAGE. And these are both Western blots, and we're looking for different toxin proteins. And we can see them here in these crisp bands in the SDS-PAGE gel. Then we can look over here and see them in native PAGE, and we can see, well, again, we're looking at the DPH2, the toxin 2 protein. You can see that depending on how much is there will tell us where ultimately it ends up here. And you can see there's a complex up here with the toxin 1 protein, and that's what we're detecting here by native PAGE. Okay, so what do we do in this coming up week for our SDS-PAGE lab? So you're going to take bacterial culture supernatant proteins, more on those later. Your standard, you're going to run a protein standard. Again, it's our old friend bovine serum albumin, or BSA. You're going to take a molecular weight marker and you're going to run these on SDS-PAGE gels. So first what we're going to do is we'll separate the cells from the spent culture broth. That culture broth is going to be our supernatant here. And our supernatant will measure the protein content. We'll set up the SDS-PAGE apparatus. You'll use these precast polyacrylamide gels. All you have to do is take them out of the package, put them in the gel tank apparatus, add the gel loading buffer to the apparatus, or sorry, to the protein samples. The loading buffer will contain, incidentally, your dye and SDS as well as a denaturing agent. So usually we'll do that in a 1-to-1 ratio here. Heat the samples to almost boiling for 5 minutes. load about 20 micrograms of protein sample per well in the gel. And then in a separate well, add 5 microliters of molecular weight ladder. Turn on the power source, run the gel. Once you've done that, stain with Kumasi blue and then destain. Then take a beautiful picture of your gel. Hopefully you'll be able to get all of this done in the time allotted in lab, and there will be an awful lot of waiting as the gel runs. Then you could do some homework or what have you and come back later. But if you do run out of time and you have to go to a different class, lab's over, your TA might need to help you with this. So depending on time constraints, but Jayashri can enlighten you as you do the lab that particular day. Hopefully we can get it all done, but just in case, we have a fallback position. Alright, so here from last year is a sample gel. It's a pretty good gel, I think, overall. 2 groups were able to fit on one gel and share one molecular weight marker here. So essentially what we did was we looked at protein supernatants. So we spun out cells of E. coli, Ralstonia eutropha, and this other Ralstonia species, and basically took a look at the proteins that were in the supernatant. We also ran bovine serum albumin in here as well as sort of a test next to the protein marker. And we were able to see distinct banding patterns depending on whether the sample was E. coli, whether it was Ralstonia eutropha, or the other Ralstonia species. We saw different bands. Some were very thick, which indicates that they're very there's a high concentration in there, and others were less concentrated. Okay, so essentially what we will be doing, PAGE gels, these SDS-PAGE gels will separate proteins based on the protein detection method that you already are familiar with, the Bradford method. Kumasi is what will detect your separated proteins. Our SDS-PAGE will separate by size only, not by charge, because we're covering these proteins in negative charge. We talked a lot about 2D PAGE, which separates both by charge and size. Native PAGE will separate based on size, charge, and shape. Okay, so make sure you are in your respective labs on time at 11 o'clock these coming up weeks, and I will see you the following week.